Great, thank you. And thanks to ASCO and Pfizer and Merck for supporting these sessions, which I think are really fantastic. So to get this session started, I'd like to talk to you a little bit about prostate cancer. I'll give you a little bit of an overview of some of the issues around prostate cancer that we've been thinking about on the African continent, as well as some of the work that we've been doing, just to give you a flavor of the kind of work that's happening, and as well some of the things that are possible in Africa. So just as a little bit of background, we have a lot of knowledge about prostate cancer incidence and mortality around the world. And we know that African descent populations, particularly those in the Caribbean and in North America, have the highest rates of prostate cancer in the world. And you can see in this upper middle section, African Americans and Afro-Caribbean men have the really highest rates. On the left-hand side of this plot is Africa in the green dots. And so while we believe that prostate cancer is an incredibly high incidence and disease, we don't think that it's got the capture of all the cases that are there. So the African situation, at least currently, is that we have relatively low incidence but high mortality. And I think that the high mortality is true. You can see on the bottom right-hand side that Africa represents some of the highest mortality in prostate cancer around the world. But a lot of that is probably due to incomplete case enumeration, meaning that we don't screen for prostate cancer in Africa, and therefore we don't detect a lot of cases. We certainly don't detect a lot of the indolent cases that you see in other parts of the world. So you can see incidence in the United States, in Europe, and Australia is incredibly high, and some of that is, again, due to screening, which we don't have in Africa. So the consequence of that lack of screening is that there tends to be a late stage at presentation. Men are diagnosed with advanced disease and have a relatively limited access to advanced treatments or even basic treatments in many cases, leading to this very high mortality rate. Not going to talk a lot about screening today, but obviously screening by PSA in the United States, for example, has been quite controversial and not completely successful. So I'm not going to imply that we should be doing screening in Africa necessarily, but we need to think about the picture of what prostate cancer looks like, why we have such late stage at presentation, limited treatment access, and what we can do, particularly in a research context, to understand how to fix things, how to make things better. Even though we have very high mortality from prostate cancer, the future prostate cancer burden is not a pretty picture. So we believe that by 2040, so in the next 20 years, we should have a more than doubling, about 115% increase in the incidence of prostate cancer. But we should have almost a 2.5-fold, 161% increase in mortality from prostate cancer among men in sub-Saharan Africa. So this is just sub-Saharan Africa, the WHO Afro region. And so we're talking about something that's not only a major burden currently, but a burden that's going to grow in the future, and is something that we need to be thinking about today, not only in terms of the clinical services and access, but also to understand prostate cancer in the African setting so that we can begin to address some of the underlying etiologies, causes, and preventive strategies that may result from this scientific knowledge we gain. What we know so far, and the next little bit of data is really is data that we have from European, North American, Asian populations. So these data don't necessarily refer to Africa, but it points out to us some of the issues that we face in understanding prostate cancer, but also the kind of data that we need to generate in Africa in order to make sure that we're dealing with risk, we're dealing with the underlying etiology of disease appropriately. So what we know is that factors that are associated with total or advanced prostate cancer risk are really quite limited. For total prostate cancer, it's age, being of African descent, family history positivity, and height. Really, none of those are really modifiable risk factors. They're factors that might identify high-risk individuals, but they're certainly not going to help us prevent prostate cancer aside from identifying high-risk people and intervening in those in some way. For advanced prostate cancer, obesity is a known risk factor. And so there's some hope that we might be able both to understand the etiology of prostate cancer through understanding obesity and the role of obesity in inflammation or metabolism or whatever the factors are that are associated with causing prostate cancer, but also it's potentially modifiable. Obviously not something that's easily modifiable, but something that we may be able to address in some way. But this doesn't really give us a lot of material to work from. It's not like breast cancer or colon cancer or some of the, and lung cancer, the other major cancers, for which we really do have some modifiable or at least identifiable risk factors on which one can intervene. So that makes it difficult for us to address how we're going to deal with prostate cancer, developing interventions, et cetera. And it also points out even more so our need to develop screening strategies that are risk stratified or risk appropriate in the setting of the African continent. We do, however, have a lot of information these days about the genetics of prostate cancer. And we know, for example, that there's evidence for association of high penetrance genes with prostate cancer, particularly BRCA2, but also BRCA1, some of the DNA damage mismatch repair genes, and HOXP13. So there are a few genes that have been pretty clearly identified as conferring susceptibility to prostate cancer, some of which have guidelines for PSA screening, some of which have less information, but may still be promising. We also know that there are a lot of low penetrance genes that confer a small bit of risk when mutated. We have at least 167, it's probably over 200, depending on how you count them, loci that are associated with risk of prostate cancer from genome-wide association studies. And these so far explain about a third of familial relative risk. So we're explaining some of that family history by genes we can measure, the family history that's associated with prostate cancer risk. This is helpful, but it's been limited so far. We have very few novel single nucleotide polymorphisms that affect African descent populations specifically. There are a couple of genes that appear to be very important for African descent populations. But what we have found, and this is data that we, on the right-hand side, that we presented a couple of years ago from our group, is that some SNPs seem to have different effects on prostate cancer risk in Black versus white men. And this is African descent versus European descent. So these are data that are relevant to the African continent. What you can see on here on the left-hand side is there's a long list, which I'm sure you can't read, of all of the SNPs that have been associated at that time, incontrovertibly associated with prostate cancer risk. And so this is a subset of all of those that we know of now. But you can see they're ranked, whereas the SNP at the very top of this list is associated with the greatest risk of prostate cancer in Africa, and the SNPs at the bottom of the list are associated with greatest risk of prostate cancer in European descent individuals. And so what you can see is that, again, this is data taken from a very large publicly available databases, plus data that we've accumulated in Africa. And we've made these models to understand, are there differences in risk based on genetics? And we do see some evidence that there are a few SNPs that are going to be much more important in terms of conferring risk to African men, and others that will be much less so. So we're beginning to get a picture, although we don't have really good data yet about the underlying genetic distribution of risk. We're starting to get a good picture about how the individuals who are African in descent might be at differential risk for prostate cancer, depending on the genes that they've inherited. So why is this important? And I'm going to focus the rest of this talk on sort of genetics and discovery, because that's an area I think we have both the opportunity to make important scientific strides, as well as contribute to the information about risk and etiology of prostate cancer in African men, so that we can begin to identify risk strata to use in screening and treatment and other kinds of activities. And in addition, to be able to develop the scientific capacity to do this work in Africa itself. So why do we want to think about genetics as a role for the prostate cancer and the management of prostate cancer in populations? Well, the first point I want to make is that we know very well now that in order to do genetic assessment, to do studies of genetics in etiology of cancer, you must have diverse research data. And there are a lot of pieces of data that illustrate that, but let me just present one to you. This is a slide from a paper done by Zach Kohani and his colleagues a couple of years ago that show an example of why diverse research data is critical for disease management for all populations. And so what you see on the right-hand side is a plot. And this plot is, these data are related to hypertrophic cardiomyopathy. So not cancer, but the same principles apply. Cardiomyopathies are hereditary and are thought to be genetically caused in a lot of situations. And in fact, you test for genetic susceptibility to cardiomyopathies, and these genetic tests inform management of disease. So here, what we have is in the blue bars, you see the frequency of some of the mutations that are shown here in white Americans. So Caucasians, European descent individuals. Many of these were thought to be really rare. You can see TNNI3, MYBPC3 genes that were thought to be incredibly rare. And when they were tested for and these mutations were found in individuals, they were thought to be disease causing. And people were treated as if they had a disease causing variant. Well, what this group did more recently is looked at Black Americans, African descent individuals, and found that in fact these mutations are pretty common in Blacks. And when they followed this up, they realized these are just common variants. They're not really disease causing. But we didn't know that until people started studying diverse populations. Until Black populations were studied, we thought these were disease causative rare mutations that needed to be followed up on. So the point here is that by studying diverse populations, you learn something that improves the clinical value for management of disease for all populations. You don't just help those minority populations, the Black populations, you help all populations by having diverse data. So this is going to be a critical feature of what I'm going to talk about next, informing genomic. It will inform genomics for minority populations. It'll impede the creation and exacerbation of disparities, and it'll avoid misdiagnoses. And one of the problems, of course, then is this data that we have from Martin et al. Last year that shows there's really, to date, very limited racial and ethnic variation in genomics research. You can see the African bar. This is over time, the number of individuals in genetic association studies. You can see almost the red part is Europeans, very much the dominant group. But the purple part is Africans. And you can see that the African bar in that plot is very small, as are all other minority groups. So we need to fix that. We need to think about how to deal with it so we improve our understanding of etiology and health for all. One way we're doing this is through a network that we've created called the Men of African Descent and Carcinoma of the Prostate Network, MADCAP. MADCAP includes about a dozen centers in North America and a dozen centers across Africa, many of which are funded by the NCI. We have some very active research going on and we're not doing just genomics. We're doing diet and nutrition and survivorship and clinical screening and management questions. But I'm going to just talk a little bit about the data that we've generated in genomics to give you the picture of what kinds of things are being done and can be done. So here's a first piece of data that I want to show you. And this is a very complex slide, of course, but it relates back to the picture I showed you before about genetic variants associated with prostate cancer. So what we did is one of these examples is we asked, well, is there some evidence of some of these genes being impacted by selection, by evolutionary forces that may have an impact to help us understand why genetics may be different in Europe versus Africa, why the risk associated with a particular genetic variant in Africa may be different than that in some other part of the world. So you can see on this plot on the y-axis is selection in Europe. That is to say, is there any evidence that something happened in European individuals that modified the frequency of these genes associated with prostate cancer? And on the x-axis is the same part of selection for Africans.
Hi,
Selection.
sorry, I mean,
And so you can see that there's one SNP. I'll just give you the example here, RS7584330. This is a SNP that's associated with prostate cancer. And something happened to it when it passed from African populations into European populations. Some selective pressure impacted its frequency, its conference of prostate cancer risk when it migrated in through populations out of Africa into Europe. Why would that be? What does that have implications for? So just as a simple example, we found that this SNP makes a disproportionately large contribution to prostate cancer risk in African men. And we have some evidence that this might be due to a concept we think of as called hitchhiking. So we found that this risk allele, the prostate cancer risk allele, is about 50% higher in Africans versus non-Africans. But as I said a second ago, it's under positive selection in Europeans. Interestingly, when we sort of looked around that region, what's going on, we noticed that this gene, this prostate cancer gene, is adjacent to a gene called melanophyllin, which is associated with pigmentation dilution. So what we think happened is that there's a prostate cancer gene, but it's next door to a pigmentation gene. And as populations moved out of Africa into Europe and became less pigmented, they ended up, the pigmentation gene got selected against, the dilution of pigmentation was selected for. And we think that, the haplotype that was protecting against prostate cancer just hitchhiked along with this pigmentation gene to high frequency in Europeans. So the way that looks is, if this is the chromosomes and we have these pigmentation gene for dark pigmentation versus light pigmentation, on the same chromosome we had a prostate cancer risk allele, and then on the other chromosome we had the reference allele that doesn't confer risk. What happened as there was migration out of Africa is that the lighter pigmented individuals carried this chromosome associated with lower prostate cancer risk, while the darker pigmented individuals who remained in Africa had kept this risk for allele for prostate cancer. So again, just one fairly parochial example of what might be going on evolutionarily, population genetics wise, that might help us to understand why prostate cancer risk and the genetics of that risk may differ in different populations. And we're pursuing a lot of these different avenues to try to understand why we think that there might be evidence for increased risk in Africa, as we have seen from the epidemiology. In order to develop this further, we created a tool. This is a chip, an array called the MADCAP Africa array. And I won't go through all these details except to say that in order to do this research, in order to be able to fully understand the distribution of genetic alleles, about risk distributions genetically in African populations, you have to build the tools that are required to understand African populations. Most of the tools that we had before, genetics, the genetic platforms, were built around African or Asian populations. This is one of the first, and really the first, cancer-specific panel built around Africa. And so again, without going through all the details, we built this panel so that we could study Africans, have the African genome well captured, but also incorporate into it loci, cancer risk genes, things like that, that we would be able to study in Africa specifically and understand the African genome. This is something that was done collaboratively through the MADCAP, but also with Afrometrics, the Center for Inherited Disease Research, Georgia Tech, and the Center for Proteomic and Genomic Research in Cape Town. So it's publicly accessible. Anyone who wants to use this now can obtain it through Thermo Fisher Scientific. So what did we do when we have this SNP, this chip? Well, the first thing is that we have an array that reveals the population structure in Africa and shared genetic ancestries among African study sites. So we can actually, using this chip, pretty much say we have genetics across Africa, and our chip captures who they are. So we can distinguish someone who is Senegalese versus South African versus Nigerian based on their genome. And that will help us then to understand both the populations that we're studying, but also use this chip to better understand cancer risk. So here's an example of what it looks like when we try to study cancer risk in African populations. So as you know, there are quantities called the polygenic risk scores. These are the combinations of genes that were found by the association studies, like what I showed you earlier. the genes, the low penetrance genes for which we studied, for example, in the data that I showed you a moment ago. When you add these polygenic risk scores up, you get a distribution of genetic risk. And you can say a population or an individual is most likely to be of higher risk if they have a certain combination of these mutations, or at lower risk if they have another combination, et cetera. Using the SNP chip that we developed, we compared what happens if you use this information in European populations versus in our African populations. So what you can see, first of all, is that this gray distribution is the distribution of genetic risk based on this polygenic risk score in Europeans. So if we use our chip and ask what is the distribution of prostate cancer risk in Europeans, you get this gray distribution. And you can see that risk is substantially shifted to the left compared to all the others. And the others are the risk distributions in Ghana, Senegal, Nigeria, South Africa, et cetera. You can see that their risk distributions are substantially shifted to the right, that is higher risk based around the genetic susceptibility that we've studied. So just using something that is fairly straightforward, a polygenic risk score, using our SNP chip that was based to better understand the African genome, we can see that there are some substantial underlying differences in prostate cancer risk compared with Europeans. And there's a lot more work yet to be done here, but I think we're beginning to start to understand that those epidemiological data that suggest high risk in African populations are probably correct, aside from the limitations I mentioned to you about the ascertainment of all these cases, but the African Americans and others who have very high risk, this might have an underlying genetic basis. And that's interesting because, of course, we think it should, based on how well we've studied the genome so far, and how much we know that genetics is important in establishing prostate cancer risk. So in order, again, back to the notion of creating infrastructure and resources and networks to make this happen, we've also tried to develop a cancer genetics network to study some of these activities and to build the translational clinical activities that will help us to take the knowledge that we derive from the African genome and put it into clinical practice. So one of the things that we've created is the African Oncogenetics Network, the AON, which has been jointly led by the Dana-Farber, by the World Economic Forum, and by BioVentures for Global Health, BBGH. And as you can see here, we have a number of centers across the US, in the UK, and across Africa. And the idea is to build up a network of centers that have the capacity to do genetics and genomics research, and then to translate that knowledge into clinical practice. So the protocols that we've begun to develop are shown on this slide. We're setting up a network for genetic testing, genetic counseling, and management. And we're starting with breast and prostate cancer. Because those are two important areas that we know are relevant to clinical management, but also very important for genetic risk. So there are a couple of activities that we've been focusing on have been to develop the identification of who could be tested in Africa, to create protocols for genetic counseling and genetic testing. And to do that, our first initial set of activities are to develop or understand what's available in terms of risk models and protocols and what patients are out there and how to ascertain them, but to develop the protocols for genetic counseling, human subjects, genetic testing, mutation curation, et cetera. So we've been spending a lot of time working on those aspects. But it's really important that while we develop these immediate genetic testing, genetic counseling protocols, we think about what the downstream activities are going to be. So with knowing genetic information might be useful, but it's going to only be useful to the patient and the physician if we also make them actionable. So we also need to identify the tools for preventive interventions, for monitoring individuals at genetically high risk, and for diagnosis and treatment. So we are also working on identifying preventive surgery, screening, and the other therapeutic activities that are specifically related to genetics, the genetically high-risk individuals. And again, many of the mutations that we talk about, like BRCA1 and BRCA2, have direct implications for the management and treatment of some of these cancers, including prostate cancer. And so we can't develop the oncogenetics testing and counseling piece without having the complete picture in place. And so that's something that we're trying to work at. Again, this gives us not only the ability to develop pilot studies to understand genetics and to do the genetics studies that are required to understand cancer in Africa, but also to translate this information, develop the infrastructure, and really leverage all kinds of strengths that are already there in Africa. And just to say, we're doing this pilot in Rwanda with a team of people who are really just fantastic, and we've gotten an NIH grant to start this going. So we're going to be developing that over the coming years. So let me just end by talking a little bit about what we need to do, summarizing what we need to do. So we need to study the global distribution of disease and its etiology so that we can provide insight into the etiology and translate that clinically. So I focused a lot on genetics here because, at least in prostate cancer, that's a very promising area. But obviously, in order to address disease, we need to have better knowledge of the epidemiology. We may need to have better understanding of tumor phenotypes and tumor molecular signatures. We definitely need to have better information and systems for access to care, access to treatment, access to screening, things like that in the larger healthcare structure, which I didn't talk about at all today, but is critical for us to be able to impact on cancer mortality. Right now, we haven't done very well with that. Most of the studies that are out there have limited representation of these populations, and diversity will not only teach us things that will help everybody, but is really required for us to develop these tools in Africa. And there are a lot of opportunities for these cancer research collaborations. There are a number of consortia, including MadCap, who's always interested in bringing on people and collaborators to help do this work. But there are many others in breast cancer, esophageal cancer, liver cancer, colon cancer, et cetera. So these kinds of things that I'm talking about have broad-ranging potential for developing and translating research into clinically and public health relevant activities. So let me just end by acknowledging some of our funding. The madcapnetwork.org is our website, and you can find more information about MadCap there. And I appreciate your attention.
So firstly, thank you. Great talk. Really interesting, and actually some of the relationship between genomics and biology. I wanted to ask you, and I actually questions either in the chat or you can raise your hand. Genomics has helped us figure out, for example, and a lot of this again was done at Dana-Farber, but the PARP inhibitors in ovarian cancer. What do you see as the future of genomic-driven treatment of prostate cancer? How are you going to translate some of what you're finding into the clinic?
Yeah, I mean, it's something that's happening today. As you know, prostate cancer is already part of the oncology clinic setting at Dana-Farber and many other places around the world. We do genetic testing for prostate cancer. We use that information to understand how to monitor risk, risk stratify kinds of screenings that will go on, but then also for specific treatments, as you mentioned, PARP inhibitors and others, so that are precision oncology tools. So that's not our future, that's our today. But how do we do that in Africa? And how do we make sure that we can make something, improve public health, improve clinical management? Well, first of all, I think that Africa should have all of the tools that we have in Boston to deal with the problems of health and to improve clinical outcomes and improve practice. Obviously, there are a lot of barriers to this right now. The resources for genetic testing and counseling, the resources for obtaining the oncological therapeutics, the precision therapeutics, they're very limited right now. But I think it's imperative that we make sure they get implemented into the African setting. And we won't do that unless we can start developing the practices in a research setting that lead us into making these clinical tools available to all. And so it might be hard to imagine why we would want to do precision medicine when there are people that don't have safe drinking water and are dying of malaria. But if we wait until those problems are solved, we will be behind, and Africans will not have the clinical, intellectual abilities to do these things in a really public health relevant way unless we develop them right now. So they just have to be done, and we have to move forward with it.
Thank you. Makes me think also that, again, in fact, let me propose it as a question, but given that the biology of prostate cancer may be different, the genomics may be different, does that have implications in terms of the public health approach? And we're going to talk later on about a risk stratified approach.
Yeah, absolutely. So I think one of the things we learn in the United States through PSA screening, for example, is that PSA screening helps some men. It certainly identifies cancers early when they're more treatable, and they can go on to have a lower mortality and lower side effects and things like that. But prostate cancer PSA screening is not something that's helpful to everybody in the entire population. So it's not something that we probably should be doing for everybody all the time. We need to stratify those individuals who are most likely going to benefit. And so that not only maximizes the outcomes for men, but also minimizes the side effects that don't need to be experienced. That kind of lesson has to be taken to Africa if we're going to screen. And the other side piece to this is that African resources for screening may be limited, maybe inherently limited always. We may not want to screen every man and follow up with biopsies and treatment all of the cases that are out there, particularly the ones that aren't going to kill them. And so we really need to figure out how to do this work more efficiently and cost effectively, identify pathways to access. And one way to do that is through this genetic information, which is really it's easy to do now. I know that it may not seem like it, but it's really easy to test people. And most of the centers that we work in our MADCAP network have sequencing equipment, and they have laboratories, and they can do this. We may decide we need to do it through panel testing or something through a company, but it is certainly something that can be done now, and it will make the process more efficient and maximize health status if we can do this effectively.
I got a couple of questions that came by email, but actually I'm going to jump ahead to one of them. Complex question, but they actually said, how much does it cost? In ideal circumstances, what would be the lowest cost to look for genetic predisposition?
So in the United States right now, the average test cost for a panel test for genetics is about $250. That's what it is in the United States. That's obviously not a realistic number for the African setting. But there's no reason why it has to be at that level. The panel that we developed for the MADCAP network, which is again different than a clinical panel, is $33. And so we're not talking about something that we may want to implement on a wide scale today, but we can do whole genome sequencing for $350 now. It used to be, well, a few years ago, it was $1 million for a genome sequence. That's and right. Thousands of dollars for a panel test. These costs are going to come down. And so one of the things we've been doing is we've been in discussions with many of the genomics companies to try to understand how they can make their services accessible to the low and middle income areas. So I think that's just a matter of time. Right now, it's not really feasible. Soon it will be, I think.
And before I've got two other email questions. Before I do, any other questions from the participants.
So Fidel has a question. What do I tell a patient with prostate or breast cancer with the BRCA1 mutation who has progressed past other lines and reads about the use of olaparib? So yeah, so obviously you're all well aware that somebody who's got a BRCA mutation is a candidate for use of olaparib. Obviously, to the degree that olaparib exists in your setting and is affordable, you should use it. But I think your question really relates to, we don't have olaparib and or it's too expensive for us to use. What do we do? And that's really, I think, the reason that we need to do this work and make linkages to the companies who have these drugs available, because they're out there. We don't have access to them. What do you tell somebody who you know there's a treatment, perhaps a curative treatment for, and you can't give it to them? That's unethical. And so right now, I don't have a good answer for you to what you can say, because it's a cost and access and supply chain issue. But it's certainly something that we shouldn't, in the future, if we work at this and do it right, we shouldn't have to tell people that they can't get access to this drug. I wish there was a better answer for right now.
Okay. So I've got another one. This was an email question. It basically says, let me see if I can paraphrase it. Biology of prostate cancer varies. What do we know about the correlation of genomics with biology? I mean, let me give you an example. We use Oncotype in breast cancer to predict prognosis and also to give us an idea about treatment. What do we know in prostate cancer, that same type of correlation?
Yeah, so I mean, prostate cancer doesn't have an Oncotype DX in the same way that breast cancer does that guides treatment. There are panels out there that say they do that, but I wouldn't say that they are necessarily something that is as actionable in the same way that it is in breast cancer. The work that's happening right now is really around, it's where breast cancer was 20 years ago, where we're defining the subtypes of prostate cancer, and we're trying molecularly, and we're trying to understand, are some of these subtypes more or less treatable by known agents, or can we discover agents that might be used in treatment based on molecular subtypes? So it's pretty much exactly what breast cancer did a couple of 20 years ago, when they started understanding that there are subtypes of breast cancer. You can define them molecularly, you can develop tests that identify those subtypes, and then you can treat those subtypes with specific agents. Prostate cancer is sort of at the beginning of that process, but I think it's headed in the same direction. And I think it's also one of the reasons that we've had so limited success in figuring out the best way to detect and treat prostate cancer, because we've been treating it as one disease, when clearly now a lot of the literature tells us it's many diseases, just like breast cancer is. So I think that we're making our way, but much more slowly than other fields have done. And if I could just follow up, Fidel also wanted sort of followed with his question. So should we test if we can't treat? So that's a really important question, because I think one of the issues when you think about genetics is wonderful if it's actionable, if you can do something with the information. If you have the information and you can't do anything with it, what's the value? Why do it? And I think that's exactly the point that we need to think about, is we need to develop the infrastructure for testing and counseling so that it's there, but we have to at the same time build up the infrastructure for appropriate screening, appropriate treatment, and whatever it may be, because knowing this genetic information is not necessarily that helpful, and in fact can be harmful if you know that you can't do anything about it. So the point that Fidel is making is absolutely right on. We have to be able to act on this genetic information. But there are other ways of acting on the genetic information. So for example, you may be able to identify people at risk, even though you may not be able to treat an individual. You can test their relatives and they identify their risk and screen them and detect early, and things like that. So there is value in genetic information, even though it might not be directly linked to a targeted therapy. Those are decisions that the clinical setting, the health systems have to make. And Rose Anorlu says, are there any plans for genetics of ovarian cancer in Africa? And the answer is yes. We are doing breast and prostate, but there are obviously ovarian cancer is very much linked to the same panels of genes that are associated with breast and prostate cancer, BRCA1, BRCA2, and a few others. So the same principles are being worked on in ovarian cancer as well, much earlier though. It is a much earlier process than in breast cancer or prostate cancer.